All right. Hi, everybody. Thank you so much to Bukenya for this invitation. I'm excited to be here. My name is Andrew Savinov, and I'm a postdoctoral fellow at MIT working in the lab of Jean-Wei Li. And I'm also co-mentored by Stan Fields at the University of Washington. And today I'm really excited to share with you our work on the massively parallel discovery of genetically encodable inhibitors against any protein. In my work, I'm really fascinated by the relationships between sequence, structure, and function across scales in biology. And by that, I mean both how sequences of biological polymers fold into specific three-dimensional structures, which then impart various biochemical functions, but also then the higher order functions that emerge from the consequences of these molecular interactions and the interactions between multiple molecules ultimately for the fitness of the organism and for organismal function. So today I'll begin by briefly telling you about my graduate work, where I focused on the molecular biophysics of RNA folding and the sequence determinants of molecular function. So I'll talk both about my work, which I did during my PhD at Stanford with Steve Block and working as well with Will Greenleaf, both my work on understanding the relationships between RNA folding and catalysis, and then understanding the roles of every residue to determine the sequence to function relationships in RNA. I'll then spend the majority of the time telling you about my postdoctoral work and my ongoing work on protein interactions in living cells and how we can understand and control them using peptides. So during my PhD work, I was really fascinated by the RNA sequence structure function relationship. And I attacked this problem using two complementary approaches. One was single molecule force spectroscopy using optical tweezers, where we actually captured single molecules of RNA between polystyrene beads that were trapped using high intensity lasers. And you could then measure with very high precision the folding and unfolding and the whole energy landscape of folding, as well as catalysis of single molecules. And I'll show you how we use that briefly to understand the relationships between folding and catalysis. And we then looked at the same model RNA system, which turns out to be a genetic switch RNA that also acts as a ribozyme and controls cell wall synthesis in bacteria, to understand its sequence function relationships using a massively parallel biochemistry approach on an RNA array. So in the single molecule side of this work, we trapped single molecules of this ribozyme riboswitch RNA system, which is a nice model RNA. And we found that actually we could characterize the full folding free energy landscape from the fully unfolded single-stranded RNA to the fully folded RNA, which is catalytically active. And by characterizing the energy landscape, we found that the key folding transition that allows for function is the formation of a single duplexed region, this P2.2 element in blue, and that element is only marginally stable, so it's barely stable. And contrary to the prevailing model at the time of how ribose switches, these RNA switches work, there was absolutely no stabilization of that folded state by the ligand that actually is the on trigger for the switch. So the ligand binds, but it doesn't change the kinetics or thermodynamics of the folding at all. This element remains only marginally stable. And by looking at the force dependence of both the catalytic functional activity of this RNA and its folding, where we could sort of bias to make the system more or less folded using the applied force on each molecule, we basically found that you can actually explain what's going on in the system fully by the force dependence of the folding, which means that really the folding behavior and the folding dynamics fully control the ability of this RNA to perform its biological function. And so, for example, if you want to target an RNA switch like this, our results suggest that you actually would want to target the folding if you want to develop an RNA, a riboswitch targeting antibiotic, for example, in this case. We also wanted to understand the sequence function relationship using the same model RNA. And the single molecule measurements are fantastic in terms of being very high resolution and getting you to the real molecular biophysics of folding, but they don't scale well to that scale. And so we opted to use this high throughput biochemistry approach with Will Greenleaf, where we actually would make libraries of RNAs, of different variants of this RNA, on a high throughput sequencing flow cell and then directly perform biochemistry on the flow cell so that we could look at all of the single and double mutants of this ribozyme riboswitch RNA in parallel. And so when we did that, we basically sequenced first using high throughput sequencing, and then on the same flow cell where we now know what variant is at each position and perform the biochemical measurements of the catalytic activity, we found we could map out the catalytic effects of every possible mutation at each site and at each pair of sites. And this is one way to look at those data, which is a heat map of the cleavage rates effects of all single and double mutants. And you see that there's, of course, a very rich structure to these data, and we analyzed it in a number of ways. But for the interest of time, I'll just focus on one thing we did, which was actually a double mutant rescue analysis. We found that if we looked at the ability of every second site mutation to rescue every single point mutation, then we could really reduce the complexity and find specific features that popped out of these data. And it turns out that a subset of these features, actually these diagonal elements I'm highlighting, fully map out the secondary structure of the ribosome. So one interesting result was that even knowing nothing about the structure, you could map the structure of an RNA of interest purely using measurements of its function by looking at all of the mutants and measuring how each affects mutation, each affects catalysis. And what we found is that we could both find the catalytic and the structural roles of each base pair in the ribozyme riboswitch in this way. And looking at the other elements, I'll just briefly go into this, and you can check out the paper if you're curious to see more. We found that we could find all sorts of other interactions that revealed other features of both the ribozyme structure and how it can change during evolution. So, for example, we saw that we could map out known tertiary interactions, but also previously unknown tertiary contacts that seem to be able to form when the ribozyme is mutating. And perhaps most strikingly, we saw that there's a lot of propensity for this same switch to actually rearrange its secondary structure in different ways, for example, moving around a bulge or shifting around a base pair to maintain catalytic activity. So this is suggestive about the different evolutionary pathways that RNAs like this can take in order to maintain the same catalytic architecture while actually evolving their sequence and ultimately potentially evolving new functions. So moving on from my graduate work, going into my postdoc, I was really excited to understand the consequences of the molecular biophysics of these sequence structure-function relationships in living cells. And cells are, of course, a very crowded and complex environment. For example, if you consider a single protein, bacterial cell division protein FTSZ, which we'll return to later, even if you just look at a subset of its interactions with other molecules in the cell, you get a really complicated network. So I really wanted to understand how we could go from these molecular understandings of how the sequence, structure, and function of these molecules are intertwined to understanding what's really going on at the higher level in the ultimate biological consequences for the organism. And as I alluded to with the case of FTSZ, one really universal manifestation of these sequence, structure, function relationships in biology is really the structural interactions of multiple proteins. So multiple proteins can come together into different complexes or more transient interactions. And it's often these interactions between proteins that ultimately give rise to higher order functions. One great example in the case of FTSZ is that this cell division protein actually needs to form filaments that then form the Z ring that contracts to perform bacterial cell division. So we're very motivated to understand how these interactions between proteins play out in cells. A great way to understand protein interactions is by perturbing them. And a fantastic tool for doing that is provided by peptides. Peptides provide a really powerful tool to both understand and control biology, because peptides can bind to specific target regions on a protein and then modulate its structure, functions, and interactions. This is becoming clear both on the side of the biology, where increasingly we're realizing there are many biologically encoded peptides with crucial functions. For example, more classically, there are many hormones and peptides that act as antimicrobials and venoms. And more recently, it's also becoming clear that there's this large so-called dark matter of the proteome of many peptides that were previously uncharacterized as gene products that are actually being made and are functional in cells, and they have roles even in disease, such as cancer. And on the protein design side of things, as you're all no doubt aware, it's becoming increasingly possible to design novel peptides or mini proteins to bind to protein targets of interest. And there's a lot of exciting possibilities there for developing targeted therapeutics against your proteins of interest. And so one really fascinating class of peptides that I've been interested in are fragments of full-length proteins. These protein fragments have some inherent capability to actually bind to the native targets of their full-length proteins. And if they do that, they can potentially bind to the correct target, but lack other features of the full-length protein, and in this way act as an inhibitor. In the classic biology sense, this might be called a dominant negative inhibitor. And so I was really interested in the ability of small peptide fragments of proteins to perform this kind of function. Historically, this idea has been developed largely through naturally occurring truncation mutations and splice variants that can lead to proteins having, or variants of proteins having a dominant negative effect. But I wanted to see how generalizable this was and to what extent just small peptide fragments of a protein could actually do this and provide us with a tool to both study and control biology. And this would really be enabled by an approach in which we could actually make high throughput measurements of many or even all of the fragments of a protein and their ability to inhibit its interactions in parallel. So I'll tell you today about how I've developed approaches to do just that and to both perform massively parallel measurements. Sorry?
Yes, Doctor, I've gotten a question around there.
Yes, please go ahead.
Yes, about protein fragments and how you're saying they act as inhibitors to the original protein itself. My question is really about, do the protein fragments, the way they emanate, is it as a result of, you've talked about self-truncation, do they truncate themselves from the original protein, or they are sort of induced within the system such that we have this sort of a fragmenting but inhibiting the process? So generally my question is about, can a protein itself truncate itself to form a fragment, the protein fragment, or it's just an external process? Thank you.
Yes, that's a great question. Thank you, Evgenia. So actually, I think both are true and both are of interest to us. So we are focusing on, in the work that I'll be talking about today, on sort of our ability to synthetically generate arbitrary sets of protein fragments and show their function and their functional potential, and how we can use them as tools and ways to study the biology and interactions of these proteins. Where so in that sense, we can make any fragments we want. But actually, there are also numerous biological pathways by which fragments like this can arise in cells naturally, which I think is what you were getting at, right? And indeed, we think, based on the work I'll show you today, that a lot of those naturally occurring fragments ought to also have functional consequences. And that's one of the directions we're really excited to be pursuing now.
Great. Thank you.
Thank you. So we developed two complementary ways to go after this problem. One is high throughput in vivo experimental measurements in living cells, where we actually put libraries of these protein fragments directly in cells and read out their functional consequences. And the other is massively parallel computational predictions using AI methods to actually predict the ability of these fragments to inhibit interactions by predicting their binding interactions. So first, I'll tell you about these experimental approaches we've developed to map protein interactions directly in cells. Then I'll show you how we can use these approaches to really show that protein fragments are generalizable, genetically encodable inhibitors, we think, of any protein, potentially, of any protein interaction. And finally, I'll tell you about how we've developed this high-throughput computational approach to predict these inhibitory fragments using massively parallel AlphaFold-driven predictions. So in terms of the first part, I'll first describe the experimental assay we developed and demonstrate how this approach works using the case study of FTSC. So in this approach, we begin by generating a library of DNA oligos encoding fragments tiling across the protein of interest, or indeed, as we'll see, the diverse set of proteins of interest. And in this case, in the work I'll be telling you about today, we made libraries of fragments tiling across each protein with a 1 amino acid step size. We then clone these fragment libraries into an expression vector and transform them into cells. And in the work I'll be telling you about today, this will be in E. coli. We can then grow the cells under conditions in which the parental proteins of the fragments are important for growth. So the fragments that inhibit the interactions of their parental proteins will inhibit growth. And so fragments, cells carrying fragments that inhibit these interactions should be depleted in the population over time. And we can quantify that using high throughput DNA sequencing to measure the frequencies of different fragments in the population over the course of this selection. We can then align inhibitory and neutral fragments back to their sequences of origin, and this way develop maps of the inhibitory activity as a function of sequence position, which I'm showing you here notionally. You could imagine there are specific regions that would then pop out as having high inhibitory activity, and then we could understand why peptides from those regions are actually inhibitory. So this example here is notional when I show you what the functional mapping looks like. So let me show you what the data actually look like for one example protein. Let's move forward. So in this example, I'll focus again on FTSZ. This is a bacterial cell divisional protein, which again needs to form filaments that then form the contractile Z ring needed for cell division. And when we look at the results for the inhibitory fragment measurements, the fragments tiling across FTSZ, here's what the actual data look like. I'm showing you the inhibitory effects along the vertical axis, where larger negative values of inhibition correspond to greater amounts of inhibitory activity. And then along the horizontal axis, we have the position of each fragment along the full protein sequence, from the N terminus to the C terminus. So each point here is a single 30 amino acid fragment of FTSZ. And you can see we perform these experiments multiple times, so we have also measurements of the error associated with a measurement on any given fragment. Okay, so we see that there are these characteristic peaks of inhibitory activity at specific sites in FTSZ, just as we imagined notionally. And how can we understand what these regions actually map to, what these inhibitory effects actually are telling us, right? So if we go back to the structure of FTSZ, we can see that it's made up of these monomers lined up head to tail. And if we take a closer look at the monomer structure, which I've just colored from blue to red from the N to C terminus, we can identify 3 approximately reciprocal regions that form the interface between adjacent monomers, which I've just called here sites 1, 2, and 3 on one face of each monomer, and 1', 2', and 3' on the other face. These are the sites that come together to form the filament. So if we map these structural features of FTZ onto our data from the inhibitory fragment assay, we see that actually the strongest inhibitory peak maps to this interaction site 1 needed for filament formation. And similarly, if we look at the other inhibitory peaks, they may actually map out the whole interface for filament formation, which I'm just showing you here in the context of 2 adjacent monomers in the filament. So we actually see that these fragments are mapping out reciprocal sites from either side of the interface needed to form the filament. And that's exactly what you expect if these fragments are actually able to inhibit filament polymerization in cells. We confirm that this is the case by actually purifying these protein fragments that are inhibitory and showing that they can inhibit filament polymerization in a light scattering assay in vitro. And notably, we could also understand why there's a third region of the interface where we don't see any inhibitory peaks. And that actually corresponds to this flexible region for sites 3 and 3', where other measurements have shown that this particular part of the interface likely is not very rigid, is flexible, and so is thought to contribute little to the binding energy. So we think that's why fragments from the third part of the interface also do not show up as inhibitory fragment peaks. So what we have in hand then is a method that lets us actually identify specific protein fragments that inhibit protein function, and we can do that directly in living cells in a high throughput way. So I showed you how this method works and how we can use it in FTSZ. And now I want to show you how actually this generalizes beyond just this one protein, and how we can then come to the conclusion that protein fragments are really generalizable and genetically encodable inhibitors of whatever protein you may be interested in. I'll show you how the results generalize. I'll show you how we can actually explain how well these inhibitors work across diverse proteins with different structural and functional features. And I'll also show you the connections of these results and of what these protein fragments do to naturally evolve peptides, which are sometimes called mini proteins, as well as the roles of different simple properties like fragment length in the function of these peptides. So because this is a massively parallel method, we were empowered to measure the inhibitory effects of protein fragments spanning proteins with extremely diverse structural and functional features. And we
We have
deliberately
a question in the chat.
Yes, please go ahead.
Yes, Felix. Can you ask the question? Felix is asking.
Thank you very much.
Yes,
I'm
you can ask.
just wondering, do the flexible regions have any bearing on the functions?
Yes. So in general, yeah. So in the example I showed with FTSZ, this flexible region, we don't see inhibitory fragments mapping to it, this flexible region of the interface. But in general, flexible regions can be important for function. And actually later I'll show you an example where a different flexible region of the same protein, which is actually an intrinsically disordered tail region, we do find inhibitory fragments that actually seem to map out key interactions with other proteins in the cell division machinery. So we actually can use these approaches to develop inhibitors and to predict inhibitors of IDRs, for example, or IDPs, where the flexible regions are crucial to the function. So in some cases, it seems that it's harder maybe to make a good binder of a flexible region, but in cases where we can, as I'll show you, we actually can understand the functions of these disordered regions, these flexible disordered regions, by inhibiting them with peptides, if that makes sense.
Yeah.
Thank you for the question. So because it's a massively parallel method, we're really empowered to look at proteins with a really diverse set of structural and functional features to see what actually generalizes about this approach. To what extent can you make protein fragment inhibitors of whatever protein you're interested in, right? And so we looked at a variety of proteins, and I'm showing you sort of the set here just to give a sense of the diversity of functions, for example, and there's also diversity of structures here. So you can see we looked at proteins involved in cell division, but also central metabolism, DNA replication, protein quality control, and even lipopolysaccharide transport. So they're very different proteins. And what we found is that across the board, just as in the case of FTSZ, and I'm just showing you a few examples here, in all of these cases, inhibitory protein fragments mapped to key regions of the proteins involved in crucial protein interactions. So for example, with RPLL, which is this 50S ribosomal subunit protein L7L12, the inhibitory fragments map out this really key region needed to form the ribosome stalk. And for DNA gyrase, inhibitory fragments map the region that's crucial for forming the C gate, which needs to open and close properly to prevent double-stranded breaks in the DNA that then are obviously a huge problem for the organism. And so we see that in general, across these diverse proteins, protein fragments really are pervasive and generalizable inhibitors. And so these fragments can actually readily inhibit protein interactions.
Doctor, I'm wondering what does enrichment stands for in this case?
So in all of these plots, thank you for the question. The enrichment is our quantitative measure of the inhibitory effect. So larger negative values of the enrichment correspond to greater inhibition. And it's actually derived from log of the ratio of frequencies of these different cells carrying these different fragments in the beginning, before you start the experiment, to different time points during the experiment, right? So we can look at how the frequency of cells carrying the inhibitory fragments depletes over time. And that's how you get this quantitative metric for how inhibitory the fragments are. Yeah, so we saw that these protein fragments really readily inhibit protein interactions in vivo, and they're quite widespread. So these functional fragments, if we just look across our data, about 25% of tiling peptides across these diverse proteins are inhibitory. So it's actually quite easy, we find, to make a peptide that inhibits an arbitrary protein-protein interaction. And so we're really excited about the potential of these protein fragments as novel genetically encodable inhibitors and drugs. We were also able to leverage these massively parallel measurements across diverse proteins to understand different underlying properties and features that actually drive function. So for example, one thing we noticed was that some proteins were a lot more susceptible or somewhat less susceptible to protein fragment-based inhibition. And when we really took that seriously, we saw that, well, in one extreme case with GRO-ES, which is one of the protein folding quality control proteins, right? We see that actually with this chaperone protein, there's very little inhibitory activity of fragments from throughout the protein. And in a more extreme case on the other end of the spectrum, we have DNA gyrates with very strong peaks of inhibitory activity. So there could be a lot of reasons for this, including differences in protein localization, structure, and the number and types of interactions formed, as well as the strength of those interactions and the details of the function of these proteins. But we actually found that there's a really single, simple property that can explain these results. And that's simply the concentration of the parental protein in the cell. In the way we do these experiments, we don't change the concentration of the full-length parental protein at all. We've just added another perturbation. We're having the cell express a fragment on top of that. And so we found that very cleanly, the more of the full-length parental protein there is, the higher the concentration of the parent protein, the less able we are to inhibit, looking on average at inhibitory effects of all fragments tiling each protein. And so GRO-ES actually has the highest concentration in the cell, and therefore when you express fragments exogenously at the same level as we do in this assay, that's the hardest protein to inhibit. And DNA gyres, on the other hand, is much easier to inhibit. And so is, for example, DHFR. And so we see this really robust linear correlation, which is really striking and I think really supports that the titration of the native protein interactions is really the major driver of fragment-based inhibition in cells. We estimate that about half of our inhibitory peaks can be explained by these effects of inhibiting the native interaction. We also found that these protein fragment inhibitory effects actually really nicely recapitulate naturally evolved mechanism, because of course, biology has already taken advantage of almost any idea I think that we can think of, right? And so in this case, for FTSZ, what we saw was that our inhibitory fragments map with a specific interface for filament formation, as I just showed you. And it turns out there's a very similar binding mode. Actually, I would say the basically identical binding mode that's already being used by the natural mini protein MCIZ, which is 40 amino acids long, so barely longer than our fragments, and it actually binds the exact same interface, and it inhibits filament polymerization in the context of sporulation in B subtilis. So fragments really let us actually rediscover and repurpose naturally evolved mechanisms, and you see the sort of convergence between what protein fragments can do and how that may be useful both to us and to nature, and how nature's already evolved proteins to do similar things. And we see another example of that with the case of this lipopolysaccharide transport system that I mentioned, where this protein LPTG is involved in transferring lipopolysaccharide across the inner membrane. And what we see is that there are only a few specific regions where we see inhibitory peaks. And all of these inhibitory peaks map to transmembrane alpha helices of LPTG. And what this looks a lot like, an awful lot like, is actually these naturally occurring membrane-spanning mini proteins, which similarly are just single hydrophobic alpha helices that regulate membrane proteins, often in response to stress conditions. And these have been shown structurally and functionally to regulate, for example, a potassium transporter or a drug efflux pump. And so fragments of these membrane proteins can also perform similar functions of regulating, we think, the function or the folding of these proteins. We also looked at functional trade-offs when we changed the length of protein fragments to understand the role of how long is there an ideal peptide length, for example, or are there reasons to use a shorter or longer peptide inhibitor? So for example, if we look at just the average inhibitory activity, looking from 20 to 30 to 50 amino acid protein fragments, the average activity increases with fragment length. But it's not all good news, because it turns out that when we look at a metric for the site-specific inhibition, the specific inhibition is actually optimal around 30 amino acids. And then these longer fragments actually are less specific. And you can get a picture of what that really means if you look at the data for the example of FTSZ with 20, 30, and 50 amino acid fragments. What we generally see is that as you get to 50 amino acid fragments, the whole baseline of inhibitory activity across the whole sequence shifts down to larger, more negative enrichment, which corresponds to larger negative inhibitory or greater inhibitory effects, right? And so there's more sort of non-specific inhibition from peptides in general with longer fragments, but the peak is actually not as strong anymore compared to, say, the 30 amino acid peak of inhibitory activity. And we think this actually happens because as fragments get longer, there are more non-specific ways for them to interact with other things in the cell, other proteins, other molecules that could lead to inhibition for reasons unrelated to the native interaction. And this also makes sense because the actual interaction interfaces that we are mapping out that these fragments are taking advantage of are quite short, often just 5 amino acids. And so potentially you just need enough structure to support this key interacting region, but adding greater length of peptide isn't always a good thing. You also see a trade-off where shorter fragments, in this extreme case, so say just the 20 amino acid fragments shown here, you're really mapping just this key region for the interaction and a little bit of alpha helix, right? With these shorter fragments, you generally see you're able to actually map interaction sites, map inhibitory regions more finely, but at the cost of, on average, weaker inhibitory effects. And so there's this trade-off where if you wanted to do mapping at high resolution, shorter fragments are really useful. If you want to make a really good inhibitor, it's useful to make your fragments longer to some extent. But if you want specificity, there appears to be, in the case of E. coli under these conditions, an optimum somewhere around 30 amino acids. And I did say that the 50 amino acid fragments were less specific in terms of just the general effects of making these peptides, but I don't want you to think that they're too nonspecific. What we actually see is that the 50 amino acid fragments still display this characteristic signature of native interaction titration driving the inhibitory effects we see. So just like with the blue data for the 30 amino acid fragments, the magenta data for the 50 amino acid fragments still have this clear, nice linear correlation between the average inhibitory effect of fragments and the log of the concentration of their parental protein. So you still see that, by and large, you see clear signatures that the inhibitory effects are being driven by these interactions. But on top of that, there's this baseline of more non-specific activity of these peptides in general.
Doctor,
So, yes?
about that same question. My network might be bad, but let me hope the question will get through. About the length of the fragment, and you sort of like give a caveat and say perhaps a longer fragment might be better, but we talked about here the 50 amino acids. But again, I was thinking, one, can we render short fragments? Like, do they render an advantage? But also, is there a statement you can make about the correlation of length of the fragment in relation to the possibility of inhibiting process to the parent protein? Like, what can be said about that length? Could it say like maybe not more than a half, maybe a half of the parent protein could be better? That's my question.
Yeah, no, it's a great question. So we don't see any obvious signature that the optimal length is sort of different for different proteins, but I think protein level resolution might not be enough. I think you might, as we'll talk about later in the next part of the talk, really need to get down to interaction interface resolution. And I think in general, probably the best inhibitor will be one that's just long enough to form the interaction that you're interested in and have enough structure to support its folding. And that's a question that will be dependent on the details of each interaction and the nature of the structure that's being formed. And so we found that on average, the 30 amino acids have these optimal features in these experiments, but I don't think that's going to be universally true for every interaction interface and every protein, but it is a good starting point. And then you can imagine tiling around that, thinking about modulating further from a 30 amino acid inhibitor, if that makes sense. And so one I think idea is in doing these kinds of experiments, it's worth exploring different lengths, because I think we saw some quite interesting effects. For example, showing that the shorter fragments, even though they're on average less inhibitory, let us show that even more tightly, it's really just this specific region that's driving the effects. And it's by shortening the fragments that we were able to do that, even though it may not be as good of an inhibitor on its own.
Yeah. Two more questions, Doctor. One is about there's been a lot of research within the community about small molecule proteins, and I just wanted to get your statement about the sort of like any behavior that could be different from fragments from small molecule proteins and sort of like performing inhibitory role to their small protein molecules. But also another one, this might be a little bit off, is about, is it a guarantee that fragments from a given molecule will all the time be able to bind to the parent protein?
Thanks for those questions. So for the second question, no, it's not a guarantee, and sometimes I should say, as we show in the papers, that I haven't emphasized that as much in this talk, but the interactions in question aren't necessarily homomeric. They're often heteromeric, and the fragments of one protein are really binding to its partner. We see that, for example, with RPLL, where we predict the binding actually in that case both to the homomeric partner and the heteromeric partner. So it could definitely be the native interface, but the other side, right? This seems to work equally well for homomeric or heteromeric complexes. In terms of, is it a guarantee in general? No. And I think going into this, I think one of our big questions was how generalizable is this? Or is it only very special fragments that can act as an inhibitor, right? Not just anything from any interface. And I think the answer is that there is some nuance there, which is why, as we'll talk about in the prediction method, it's not straightforward to just say, oh, anything from an interface will be a good peptide inhibitor. But there are certain properties that make them better. And one thing we're interested in is figuring out which properties make the best peptide inhibitors so good compared to other fragments that either have no effect or are only weakly inhibitory, right? So yes, I think that the devil's sort of in the details on that. It's not always true, but I do think our result that such a large fraction of tiling fragments are functional suggests that this is a lot easier to do than people had thought in the field previously, at least as a baseline of getting an inhibitor, right? And then of course you want to make the best possible one. But I think our results suggest that in general, there's a lot of functional potential in just arbitrary fragments. And then but exactly which ones are the best inhibitors does depend on the details, if that makes sense. For your other question about, I think when you were saying small molecule proteins, do you mean sort of designed binders?
Yes,
like,
yes.
Mini protein designs from, say, David Baker's lab, for example. Yeah. So, designed binders. And if I think if I understood the question was sort of, can designed binders also act as inhibitors? Is that right?
Yes. Yes, would they be able to inhibit on these other molecules being of that size?
That's right. Yeah. So I think so. And so I think there's a lot of interesting intersection with how we're looking at protein fragments as a way to kind of generalizably make inhibitors, and then design approaches which could target the same sites, right? And absolutely, I think you can design binders. And in fact, one really interesting question is, I think we've shown that one application of this protein fragment approach we've developed is to map sites. And then you could imagine targeting those sites with diverse approaches, potentially making an inhibitor with different properties that you want. You could imagine then targeting the same sites we've discovered using small molecules, traditional sort of pharmacology, or using protein design. And I think that's a really exciting future direction. And it's actually something that we're also getting into ourselves. All right, thank you for those questions. And so I think for the interest of time, I'll go through this part quickly and get to then the AI-driven prediction part of the talk. So basically, the long story short is that simple biophysical properties don't suffice to explain the inhibitory effects we see experimentally very well. The two most predictive features are actually the parental protein and the length of the fragment, which basically comes down to the protein concentration and the length effects I already told you about. So doing this kind of analysis of variance doesn't reveal any clear effects. It's not like all of these inhibitory fragments are hydrophobic, for example, or charged. It's more bespoke for every given interface. And that kind of makes sense because although the average effects can be explained by effects like protein concentration and fragment length, there's a lot of spread around that, which gets back to something I've been alluding to throughout, which is the details, the molecular details of each interface. And so to sort of get into those questions, we then moved into actually using prediction approaches to see if we can predict which fragments are actually the best binders. To summarize the first part of the talk, I'll just say that basically we found that with this massively parallel approach, we can map key interaction sites and develop peptide inhibitors of diverse proteins of interest very readily, and that protein fragments have a lot of functional potential. just for whatever protein you may be interested in. We showed that inhibition is driven by titration of native interactions in cells, and that these fragments are generalizable inhibitors, and that the fragment length effects actually matter. And so it is worth thinking about how long your fragment is, and there are functional trade-offs between specificity and strength of inhibition. And finally, getting into where we're going next, there's a lot of details of how these inhibitors work that do depend on the specific properties of interactions rather than generic properties like charge or hydrophobicity. So that brings us to the final section, where we're using approaches to predict these inhibitory fragments in a high throughput way computationally. So I'll show you our method for doing this, how effective it is in predicting known interactions, but then beyond that, previously unknown interactions, and how we can bring this all together and connect it back to the experiments by actually comparing the predicted binding modes with deep mutational scanning in cells and showing that the sites we predict are important for binding are indeed the sites that drive inhibition. So we really wanted to predict which protein fragments are good inhibitors using AI methods. And we wanted something that could work on the same scale or even a larger scale as what we can do with these massively parallel experiments in cells. So we wanted to mirror these high throughput experiments computationally. And we reasoned that AlphaFold predictions should provide a parallel way to do this, to both discover inhibitory peptides and provide support for their mechanistic interpretation, if this works as well as we hoped. And this is work that I did in close collaboration with Sebastian Swanson, who was a former PhD student with Amy Keating, also at MIT. So this approach we developed, which we called FragFold, works like this. We begin by identifying interacting proteins of interest, and we then computationally generate fragments tiling across our proteins much in the same way as we do in the experiments. In fact, we did the exact same thing, and we looked at fragments tiling across the proteins with a 1 amino acid step size, and in this case, considering fragments of 30 amino acids. But again, we can vary the length just as in the experiments. We can then perform massively parallel modeling of how these fragments interact with proteins of interest using AlphaFold. And importantly, and we can get into this if people have questions, the modeling was performed using AlphaFold 2 in its monomer mode, using the monomer weights, not AlphaFold multimer. And we did this because it is possible to predict multi-chain interactions with monomer. And the big advantage is that we didn't want what we got as the output of these predictions to be driven by memorization of the full-length protein-protein interactions, which is actually in the training set for AlphaFold multimer, but not in the training set for the original monomer weights, the original AlphaFold 2. And so we wanted to see if we could get a clean prediction of these interactions purely based on what AlphaFold has learned about protein folding effectively. And we then took these tiling predictions with FragFold and used the new metric we developed for the extent of predicted binding, which I'll tell you about in a second, and looked at the predicted contact, predicted binding level as a function of tiling fragment position. We could compare that directly to the experimentally measured inhibitory effects I just showed you, also as a function of tiling fragment position for the same proteins. And we'll briefly talk about this. We can come back to this if people have questions. But the way we actually did this is that we actually separated the MSA generation step from running the AlphaFold predictions. And that had a huge benefit for us, because otherwise this prediction speed would really be bottlenecked by needing to generate the MSA for every single tiling fragment, which takes a long time. But we can actually pre-generate all the MSAs for the proteins that we'll be putting into the predictions, and then chop up the MSAs themselves to use as inputs for the predictions. And we found that that worked very nicely. So here is an example of our results for the case of the cell division protein FTSC. We have here the predicted binding in blue from FragFold along the vertical axis as a function of tiling fragment position. And then in green, we have what I showed you before, which is the inhibitory activity in the high throughput in vivo assay, again, for these same tiling fragments. I've just flipped the axis so that now it's the negative of the enrichment, so that larger positive values here correspond to greater inhibitory activity, to make it easier to compare to the FragFold predictions. And what we see is that, really strikingly, for every inhibitory peak in green, we see a corresponding predicted binding peak in blue. Not only that, but when we look at the molecular details of the predicted binding modes, in other words, the structures that are generated by these massively parallel AlphaFold predictions, we see that the inhibitory peaks that are predicted correspond to native-like binding interactions. But notably, they're not just identical to the native binding mode. Again, consistent with the idea that we're not simply looking at memorization here, we're using AlphaFold 2 with the monomer weights, right? We see that actually the predicted binding modes are different in subtle ways that do end up influencing which residues are predicted to be involved. And in general, the fragments are predicted to bind these interfaces more tightly than the fragment in the context of the full-length protein in the experimental structure. And based on our results later from the computational scanning that I'll mention too, we think this makes a good amount of sense that a liberated peptide fragment may be able to bind in a different way that takes advantage of being able to form more contacts, right, than it does or than it in fact needs to in the context of polydentate interaction with many parts, this fragment just being one part of a larger interface. And we see the same result across all of the inhibitory peaks of FITC. For each of these cases, the predicted binding mode in blue is similar but non-identical to the experimental binding mode in the context of the full-length protein-protein interaction. So we were wondering, of course, how well this generalizes across proteins with different structural and functional features. So similarly to what we did in the experimental work, we looked across these diverse proteins, and we saw that, indeed, just as in the case of FITC, we see much the same effect generalizing across these really diverse targets. In all of these cases, there's a preponderance of being able to predict correctly each of the inhibitory peaks we measure experimentally, and that corresponds to a native-like binding mode. Overall, statistically, that corresponded to about 87% of all known inhibitory peaks being predicted as native-like fragment binders, binding in a native-like mode. And we also performed deep mutational scanning experiments where we actually looked at about 7,000 mutants of about 43 protein fragments that actually span 9 diverse interfaces. And we showed that the modeled binding residues indeed are driving the inhibition that we see in vivo. Now, what about the failures? So what for the set of interaction interfaces that we looked at where we had the known inhibitory binding peak, but we did not predict native-like binding, what was interesting is that we often saw that the predicted lack of a native-like contact actually came tantalizingly close to predicting the correct interaction. But what we saw was actually instead something non-physical. So the interface RMSD, for example, could be quite good, right? Say 3.2 angstroms. So the fragment's almost in the right place, but it's at a non-physical distance from its interaction partner, such that you wouldn't actually expect them to be interacting. So this kind of suggests that some of the failures were driven by a failure of convergence of the AlphaFold model prediction, that potentially these actually are very close to predicting binding, even in those cases that don't fit our criteria for a good binding prediction. All right, so we then wanted to go further. So we had really good success rates at predicting known inhibitory peaks that we measured experimentally, but we wanted to generalize that further and say, what fraction of all inhibitory peaks are predicted, not just the ones where we have a structural interpretation based on experimental structures. And similarly, we wanted to, so we're sort of looking at peaks from the experiments and comparing them systematically to all of the peaks from the predictions. We also wanted to go the other way and see what fraction of all the predicted binding peaks are actually inhibitory. So we did this really systematically, where we actually developed a method to call the inhibitory and predicted peaks systematically, automatedly, without human intervention, so that we are not biasing what we're calling a peak. And we also automated this approach based purely on peak overlap. So we're trying not to use any of our knowledge of how we'd interpret an interaction, just saying, are these peaks overlapping between the predictions and the experiment and vice versa? And when we do this more systematic comparison, we see that we still have a really nice success rate. So across all of the proteins, we saw 64% of all the inhibitory peaks were predicted, and about 68% of all the predicted peaks were indeed inhibitory. So notably, the numbers are lower, and that kind of makes sense. There's going to be types of interactions where AlphaFold actually makes a bad prediction, and also types of interactions that AlphaFold is not capable of predicting, such as, for example, protein folding inhibitors, which we have evidence are a subset of what we see in our experimental data. But with that being said, 64% is quite nice. It's more than half of the inhibitory peaks could be predicted. And perhaps most importantly, the 68% number suggests that, that means that if you have a predicted inhibitory fragment, there's a good chance it actually is inhibitory of the interaction. And that's really the most important number here if you're thinking about using FragFold to develop new inhibitors of a protein where you have no fragment scan data going in. So overall, these results suggest that FragFold can be automatically applied to any protein of interest. And the high success rate opens up proteome-wide applications that we're really excited to be pursuing. Now, given these results, we wanted to see if FragFold could predict novel binding modes of proteins that were not previously known experimentally. That would really be one of the best tests of, is this actually predicting interactions beyond even one that we know, and so can use this as a sort of a discovery tool and a tool for mechanistic interpretation. So as our case study for this, we'll go back to something that I brought up earlier in response to one of the questions. We looked at the C-terminal intrinsically disordered tail of FTSC. This region of the protein is known to form crucial interactions for biological function, interactions with other proteins in the divisome, in fact, that are needed for cell division. But the structural basis of those interactions was extremely unclear. So excitingly, we saw that for each of these cases with interactions with different cell division proteins, FragFold predicted novel protein binding modes for the C-terminal tail region that agreed with totally orthogonal genetic and biochemical evidence. Because people have done great work measuring effects of mutants on the ability of these proteins to interact, but we had no structural basis for how they were interacting. For example, for how FTZ tail interacts with FTZ itself, there was evidence that it was an autoregulator of the GTPase activity and filament formation. But we have the first molecular model for how that actually works, where the C-terminal fragment is shown to actually directly impinge on and enter the GTPase active site. Similarly, for the interaction with MinC, for example, there are even specific residues that were shown to be important for binding that our model predicted naively with none of this information actually being present in the training data. So even specific residues needed from either side of the interaction were indeed predicted to be used for binding. And we saw similar results for the other cases. So overall, I think our results show that FragFold can be systematically applied to diverse proteins. And this high success rate is really exciting to us, as I mentioned, because we can really think about the applications of this proteome-wide. And as an interesting vignette, we find that FragFold can predict these interactions where AlphaFold itself on its own fails. And this really highlights the benefits of modeling protein fragments if you want to understand protein interactions, even if you're not interested in protein fragments in and of themselves as functional entities, though I think you should be. And so, for example, if for this case of the C-terminal tail of FTZ, as I showed you, we can predict its functionally relevant interaction with the GTPase active site. But then if you model just the full-length protein as a monomer in AlphaFold 2, you do not predict this interaction. And even if you model a dimer of full-length FTZ, you also don't predict this interaction. And this is in agreement with contemporaneous work that came out from a couple other labs, including Katja Luck's lab, showing that the modeling of fragments lets you recover interactions correctly that you actually miss otherwise if you try to model the whole protein. And I think this has to do with the fact that AlphaFold gets too satisfied with satisfying other contacts, and it just misses some contacts completely if there are too many possible interfaces that could be occurring between proteins. So we also showed that if we looked at the detailed properties of the binding modes from FragFold, they're actually comparable to those of known previously experimentally characterized peptide binders with, for example, crystal structures. So when we compare to, say, the PepPro database, right, and we look at known binders from that database in blue versus the FragFold predicted peptide binding in orange, we could also even calculate the Rosetta binding energy. So predicting the binding energy, the relative binding energy for the fragments compared to known peptide binders. And they're very similar to the known binders, only about 20% weaker. And when we do relaxation using Rosetta, so relaxing our models, which we haven't done at all in everything I've shown you so far, they're even more similar. And you can see that here, this is just showing as well that when we do the relaxation, it doesn't substantially rearrange the predicted binding modes either. And an example of that, illustrating that more schematic, more in a little more detail, is shown here, where we can see that when we start to relax the models using Rosetta compared to just the naive starting point of exactly what AlphaFold predicts with no further relaxation, which is in green, we see that relaxation does not substantially change almost any of the binding modes. And even in cases where there are rearrangements, there are largely some rotation keeping the same interface. Finally, we found that the FragFold predictions actually exhibit good specificity. This is a major question. If you imagine, what if the fragments are binding another protein? Are we predicting the correct partner? It's especially important as you think about many possible partners in the cell, right? And we found that the specificity was really nice. So, for example, fragments of FITZ are predicted to bind FITZ. Fragments of GFP, which is not, of course, made by E. coli normally, are not predicted to bind FITZ at all. That's what's shown in gray on this top left plot, compared to in blue, the FITZ data. And similarly, we could look at eGFP fragments binding FITZ. We don't see that, nor do we see FITZ fragments binding eGFP. And eGFP fragments are predicted to bind GFP itself, which is, consistent with the dimerization of GFP. And on the other hand, we looked at many examples, right, to sort of generalize this more. So we looked at DNA gyrase fragments binding FITZ, and we saw nothing. But then in some cases, FITZ fragments were predicted to bind DNA gyrase to some extent, but notably to a weaker extent than DNA gyrase fragments and with much narrower peaks. So one thing that we came to from all of these analyses is that we can also use, and we do use, the peak width as one of the criteria for distinguishing true positive peaks that appear to reflect specificity of interaction. So we did this really systematically. I won't go into all the details, but we looked across scans of one protein against many possible partners for many cases. And overall, what we came to, doing this for a variety of cases, is that we had a metric for specificity, which is the density of peaks for on-target predictions versus off-target predictions. And it was generally on the order of tenfold. But notably, for one of the proteins where for RPLL, we actually predicted a lot more predicted off-target binding. And we actually are wondering to what extent that may reflect biologically relevant interactions that have not yet been characterized for this ribosomal protein, because some of them look pretty compelling. And there are some interesting results with the RPLL fragments where they just have a really strong inhibitory effect that makes us wonder if there's something to that. So that goes to the idea that you could perhaps use these approaches to actually discover new interactions, just as like we showed with the case of the FITZ C-terminal tail. Finally, just going back to this idea of how we closed the loop and did deep mutational scanning experiments, I previously already told you that these deep mutational scanning experiments systematically showed us that the sites the model predicts are important for binding indeed are sites that are important for the inhibitory function in cells. And we could go further, and actually one really cool result from the deep mutational scanning was that we could look at the mechanisms of loss of function due to mutation, and they made a lot of sense. So for example, looking at scans of this particular interface from FITZ with protein fragments, and again, this is in actual experiments in vivo, we see that mutations of this residue, this valine 171, to different residues are very detrimental when especially when you mutate it to a charged residue. And that makes sense because this residue seems to be involved in a key folding interaction, making the fragment into a type of hairpin that then binds to its interface. So one thing we see is that fragment folding is really important, and mutations that disrupt the fragment folding that would let it bind the interface make a worse inhibitor, which makes a lot of sense. Whereas mutating it to, say, an alanine, which is similar to valine, it is not as bad. And a larger hydrophobic actually gives you something more similar, like with leucine and phenylalanine being more similar, right, in this for forming this kind of hairpin. And we saw similar effects for a residue at an interface. Rather than in folding, it's directly involved in the interaction, where this leucine residue, if it's mutated to charged residues, totally disrupts the inhibition, which makes sense that totally disrupting the interaction, which is the hydrophobic interaction. But then you can attenuate the effect. But in this case, basically, other mutations are also not good, probably because of the way the leucines need to all fit together in the interface. But the charged residues are definitely the worst at that site. Perhaps most excitingly, we could also actually find, in a high throughput way, superior inhibitory peptides just through single mutations. So we could go beyond already these quite effective inhibitors from the naive protein fragments. And we found we can very readily discover stronger inhibitors going beyond what biology has developed as sort of these fragments of the full-length interface. So for example, when you look at this valine 262 in this fragment, it's also forming a kind of hairpin structure, which presumably stabilizes the folding of the fragment. And we see that mutating that valine to a phenylalanine, which presumably makes a stronger hydrophobic interaction, actually makes a much stronger inhibitor. And so we're really excited about the potential for really leveraging our ability to do these high throughput experiments to also rapidly develop even better inhibitors that could be useful both as tools to study the biology and drugs to actually target these proteins. So finally, just to bring us to our conclusions, today I showed you how protein fragments are generalizable, genetically encodable inhibitors of protein interactions, and we think can really be applied to any protein of interest. I also showed you how our computational fragment scanning approach, which we call FragFold, allows for high confidence prediction of inhibitory fragments across proteins with diverse structural and functional features. I showed you how FragFold can even predict novel binding modes in agreement with orthogonal experimental measurements. And I hope I've convinced you that these high throughput computational and experimental approaches are highly complementary for both discovering novel peptide inhibitors and interpreting their mechanisms of action in the cell. Looking forward, I'm really excited to leverage these approaches I've developed, both as I continue my postdoc and in my future lab, looking at these protein fragments as a scalable tool to really enable a systems biology of protein fragments in living cells. And I'm also really excited to leverage protein fragments to understand protein structure-function relationships in vivo in their natural context, using these fragments as generalizable probes. I'm also very excited about the biology of these protein fragments, really thinking about how we can use them to understand the physiological roles now and the evolutionary implications of protein fragments that arise naturally. And finally, of course, we're very excited about these novel types of peptides as a path to novel drugs, where we can potentially quickly develop novel inhibitors against diverse proteins of interest. So with that, I just want to thank a number of people, in particular, Stan and Jean, for all of their mentorship and support, and Sebastian and Amy Keating, with whom it's been really fantastic to work on these computational approaches using AlphaFold. And thank you as well to all of the members of both the Fields Lab and the Lee Lab, and to my funding, and to you for listening. And I'd be very happy to take any additional questions.
Thank you. Thank you, Dr. Andrew. Thank you. Really, a few questions from my side, but there is a first question from in the chat. I will read it for you, if it's okay.
Yes, I'm ready.
Herbert is asking, how do your experimental and computational approaches work together to identify protein fragment-based inhibitors? And what key biological principles did you discover about these inhibitors?
Yeah, so to summarize on that point, I think the synergy comes in many forms. I think one thing is that I don't think we ever would have been able to develop this computational method and sort of believed how well it was working and benchmarked it if we hadn't also had the complementary high throughput experiments, which we in fact in this case did first. Because without that to ground these predictions, I think it would be easy not to believe that these fragments really can do this, right? Even if AlphaFold predicts it. So there's even the level of, would we even pursue this if we hadn't done the experiments? But then beyond that, the experimental data let us benchmark the predictions to actually exclude some of the false positives and come up with ways to really pick out the subset of peaks that correspond to real inhibitors more faithfully. Another way that the experiments and the computational approaches are really working together here is that we can do the deep mutational scanning, as I showed you, which is, again, experimental work. And that really lets us show that indeed these residues that are involved in the context in the predictions are being used for binding in the cell. And that's really useful just practically, right, in terms of making even better inhibitors and sort of further developing, modulating what we see from the natural fragments. But you could also imagine looking forward, I think we're very excited about using our experimental data to further train a model that's even better, right? To sort of use our ability to really measure these effects in high throughput to then potentially make a pipeline that lets us get to even faster known good hits, right? And what was the other part of the question?
The other part was asking, what key biological principles did you discover about
Thank you. Yes.
these
So I think
inhibitors?
the biggest biological principle here is that fragments of proteins are generalizably able to function as binders and rewire interactions of full-length proteins in cells. And I think that's very exciting because there are a lot of ways by which fragments can arise in cells due to errors and mutations and proteolysis and so forth. And our work suggests that we have to take the fragments and small peptides that arise in these kinds of processes, like even cleavage of a protein, to process it really seriously as functional entities in and of themselves, right? And I think there's a lot more functional biological peptides out there than we know now. And we're really excited to explore that and to discover them and see how far that goes.
Okay, members, if any of you has a question, just unmute yourself and say it. In the meantime, I'll pose my question to Dr. Andrew. Doctor, somewhere you talked about, or you said that for the measure of specificity, like the number of overlapping fragments or the peak width was a strong sort of filter for specificity. And because I saw the graphs and really it was demonstrating that, but I'm trying to understand why is that the show, and probably I'm missing the true meaning of specificity in this context of fragfold.
Yeah, so it's a good question. So in that context, I was talking about specificity as to what extent is the ability of fragments to bind a target specific to what we think is the actual functional target of that peptide versus there's a few ways non-specificity could arise. It could be errant predictions that fragfolds predicting all sorts of things just bind because they're hydrophobic and they just always all bind each other. Right, that was one possibility that I think our specificity test suggests is not the case, right? The other way specificity could arise or non-specificity could arise is sort of this question of to what extent binding is in fact specific. Are there other kinds of interactions in the cell? And of course, we can't tell if they're unknown to us. We don't know if it's a false positive or not. But when there are cases where we really don't expect any interactions to have evolved, we see a very clear specificity. For example, I mean, in the extreme case, we look at GFP, which has never been around in E. coli naturally, and we indeed don't see predicted binding. So I think, and what we see with the peak width, right, turned out to be really useful. I wouldn't say we fully understand why this is, but we have some ideas. So we think that the sort of true positives that agreed with our inhibitory data also tend to be associated with a sort of minimum width of peaks. It's not always the case, but there's a correlation. And I think what this may have to do with is that if there's a really narrow peak, like in the extreme case, AlphaFold might, only predict binding for a specific fragment, but then you shift by one amino acid, you don't predict binding anymore, right? And so what I think that reflects is that the model, I should say we didn't get into this very much, but we found, that this new metric of the weighted contacts is really powerful for these predictions, where we actually take the number of contacts from the structural model and then multiply it by the confidence interval. But the confidence itself we found wasn't working very well for us for predicting these fragment binders, right? And so I think getting away from that, I think the super narrow peaks may in many cases reflect something that, AlphaFold just barely predicts as a binder, right? And even a small change of just moving by one amino acid on either side is no longer predicted to bind. That kind of makes you wonder how robust the prediction of binding really is. So it's sort of an additional metric potential additional metric for how confident we should be in a prediction. And these wider peaks mean that even if you shift, this core region of the fragment that seems to be involved in binding keeps being predicted. And we found that's associated with agreement with experiment.
Okay. Wow, this was a run. I want to thank you, Dr. Andrew. That's the end for me from my side. I don't know whether anyone in the audience has a question, the rest of the members, but if no, I want to come to the close of this session by thanking Dr. Andrew for really saving up your time and decide to share this valuable novel work with us. We appreciate as we are making our footing into this world of drug discovery and computational biology. I can't thank you enough. Thank you very much, and thank you very much indeed. And to the members who have attended, thank you also. We hope that Dr. Anytime, I know you've pushed out a lot of work, and I'm sure more is coming. We hope that once we reach out for knowledge again in the future, the response will be again expeditious like you responded this time. We want to thank you and thank you very much.
No, thank you, Bukenya. Yes, I'd love to come back. It's been great to join this computational intelligence group. So I really appreciated the chance to come out. And yeah, it was great to talk with you and with everybody here.
Thank you. I want to say, please enjoy the rest of the day. Have a good day. And to the members, a good night and a good evening here in Uganda. Bye-bye, everyone.
Same to you, and thank you.
Thank you.
All right.